Hello, everyone. Welcome to our first MDICx Series teleconference. The goal of these telecons is to engage our membership and the broader public, bringing MDIC progress to the forefront and highlighting emerging trends in regulatory science. Today, we have a wonderful lineup of speakers, and this teleconference is hosted by the MDIC Computational Modeling and Simulation Project. I'm Dawn Bardot, and I'm the Program Manager for the Modeling and Simulation Project at MDIC, the Medical Device Innovation Consortium. Today, our speakers will include Kyle Myers from the FDA. She is Division Director of Imaging, Diagnostics, and Software Reliability from the Office of Science and Engineering Laboratories. She's also the MDIC Computational Modeling and Simulation Steering Committee and FDA PI. She'll be talking to us today about mock submissions to the FDA CDRH, history, and the lessons learned. We're also joined by Tarek Haddad, Senior Principal Statistician from the Cardiac Rhythm Management Division at Medtronic. Tarek is also the chair of the MDIC Modeling and Simulation Working Group entitled Clinical Trials Informed by Simulation and Bench. Tarek will be talking today about how Bayesian incorporation of virtual patients to inform clinical trials can be done. To let everyone know, we're in presentation mode on this teleconference, so Tarek, myself, and Kyle will be the speakers, and the audience is muted. To facilitate question and answers at the end of our presentations, we'll reserve the last 15 minutes, and we'll ask that the audience type in their questions in the chat dialog box. You can access that box by pressing the green balloon at the inside of the Join Me tool, and this should open a drop-down. I've posted a couple of messages in there welcoming everyone to the meeting, and so you should be able to view that. If there's any technical challenges, feel free to drop a line in that chat box as well, and I can see if we can do anything on the fly to better facilitate the meeting. But with this, we'll get underway and start the presentation. I'll start by giving you a bit of an overview about MDIC. MDIC was founded in 2012, and the Medical Device Innovation Consortium presently has 44 members and 5 projects that are underway. Today we'll be hearing from one of those projects, the project's program manager, myself, Dawn Bardot, and one of the working groups as regards to their progress to date. The MDIC is a 501c3. We're a public-private partnership, and we're collaborating in the pre-competitive space on regulatory science, striving to make patient access to new medical devices and technologies faster, safer, and more cost-effective. And we do this through our public-private partnership, bringing together government, FDA, CM&S, and NIH, industry, including the device manufacturers and the service support and software providers, as well as nonprofits, including patients, providers, and academics as subject matter experts. And we work together to bring to bear our combined resources to accelerate the progress and ultimately achieve results, bringing those technologies to patients faster, safer, and more cost-effectively. Today, I'll be representing our Computer Modeling and Simulation Project and our progress. The vision of the CM&S project is quick and predictable access for patients to these innovative technologies, and we want to enable access through providing safety and efficacy evidence through 3 pillars: the typical bench, animal, and human pillars of safety and efficacy evidence, and also computer modeling and simulation. In the 21st century, computer modeling and simulation is ready to provide that evidence. We have great models out there, and we're working on increasing ways that we can confidently use those models. The Modeling and Simulation Project held an inaugural kickoff last spring, where we brought together our member executives and fellows, and we prepared a survey asking, what are the barriers today in realizing this vision of using modeling and simulation. And we had a variety of responses, including the expertise necessary to create and run these models, scientific maturity, regulatory expectation uncertainty, cost, and demonstrating return on investment. But by and large, by a far margin, the regulatory expectation uncertainty was the largest. So what we decided to focus on was ways to remove the barriers to using modeling and simulation at that regulatory stage in the total product life cycle. And we believe that ways that we can impact that are through mock submissions, where we create an open dialogue about expectations and what's possible with technology today. We can create tools, models, and simulations that can be reused over and over again in a variety of submissions through the medical device development tool process. We can create white papers, and we can bring together the community, creating communities of practice around regulatory grade modeling and simulation. Another thing that we learned from that survey was where some of the opportunities lie for modeling and simulation, where modeling and simulation isn't extensively used today. And the two standouts are in the clinical phase of the total product life cycle and the reimbursement phase of the total product life cycle. So I'm so excited today to be able to bring you one of our working group chairs who's bringing a team together to address one of those opportunity areas. But we don't just have one working group, we have 7, and our 7 working groups in the Modeling and Simulation Project include a neurostimulation working group, the working group that will be talking to you today, combining bench and simulation to impact and inform clinical trials, human heart and vasculature working group, orthopedics, MR-induced heating, a blood damage working group, and a working group creating the infrastructure for a library of models, data, and validation information. And with that, I'll introduce Kyle Myers to begin our first presentation today.
Great. Thank you, Dawn. It's my pleasure to be here representing the Center for Devices and Radiological Health and our significant interest in collaborating with the modeling and simulation community through the MDIC. I will be talking this afternoon about how we have used mock submissions in the past to communicate the intricacies of regulatory processes to communities that have less experience in working with us, and some of the lessons that can be learned through this way of working together, some of the lessons that we learned in engaging in this kind of process in the past. So if you could go to the next slide, please. Here's my very simple outline. I'll be talking about what a mock submission is. I'm using, as a history lesson, the example of proteomics technologies. I'll talk about why we made use of a mock submission process in that space, what the outputs of that collaborative effort were, and then I'll draw some conclusions. The next slide, please. So what is a mock submission? It's a representation of a pre-market application. Our primary pre-market applications are PMAs for primarily Class 3 products for 510Ks. There are other kinds of mock applications that might be made to the agency, but those would be the ones that I think would be most relevant for the kinds of work that our working groups are engaged in today. And by mock, we mean that it's most likely going to be a hypothetical device, something that would be a construct with hypothetical characteristics, hypothetical perhaps companion information, where that information and those characteristics are meant to probe the intricacies of our regulatory process and might be contributed by a collaborative effort, a group of folks who are looking together to gather information that would be relevant to the kinds of things that they're thinking about. And so, again, we want to keep emphasizing that as MDIC is a collaboration, mock submission is a collaborative effort that can involve multiple investigators, companies, stakeholders, and that can include, as we will find in the example that I'll be talking about, academia and other government agencies. The next slide, please. I've already alluded to the fact that, yes, CDRH has seen and participated in examples of making use of this mechanism for working together in the past. We've seen mock submissions in the case of the specific application area of protein-based multiplexed assays, where, as you'll hear as I talk about this a little more, there was an appreciation that that was a community that was at one point in time not fully aware of the regulatory issues involved in bringing new devices of that sort to market. And so mock submissions were used to evaluate what criteria and what sorts of studies would be needed to validate those devices. And you will, I believe, hear how mock submissions provided a mutually beneficial way for the device developers and our regulatory communities to identify those issues that the field should address. Next slide, please. Of course, there are many sources of possible information for vendors, investigators, especially in this modern world of the World Wide Web. We post guidances online, and we work as scientists in the agency in the development of standards and their regulations. But at the same time, we can appreciate that it can be complicated for new investigators, new developers to understand our regulatory process when we maintain confidentiality regarding test-specific discussions and submissions for regulatory purposes. And so the idea of a mock submission is to open up that kind of discussion to allow for others who have not yet engaged in regulatory test-specific discussions with the agency to learn together information about the regulatory process and requirements. Next slide, please. So this is the example that I'm going to point to as one that you can learn a lot about in the literature and help to put meat on the bones of what is a mock submission and how has it been of value to a community in the past, how we might learn from the lessons that were learned from this effort and take it into some of the work that we're doing with our MDIC working group. So NCI stood up an initiative in 2006, and it had a lifetime of about 5 years, called the Clinical Proteomic Technologies for Cancer Initiative. NCI stood this up to develop approaches to address a number of issues related to the variability from multiple sources in the development of these multi-index, multivariate assays in the field of proteomics. And I won't read them all here, but you can see them before you. And as computational modelers and simulators, we know that we all have our own sources of variability and how we develop and inform models and what we understand about what we're trying to model and how we might see concerns in verifying our models and showing that they have clinical validity. So next slide, please. In the case of this particular application area, we had a mechanism for talking with NCI that exists to this day called the Interagency Oncology Task Force, and that's a way that we can work across agencies within HHS in areas of interest in cancer. And in this particular subcommittee on molecular diagnostics, you see here members and co-chairs from FDA and from NCI. And together it was come to there was an appreciation that came to be shared that there really wasn't the kind of guidance that was needed, useful for multiplex proteomic assays. And so it was determined together to take these two action items here to address these needs. One was to convene a working group, a workshop that would bring together the community to discuss their previous and current efforts, and to, out of that effort, to develop a white paper. And I know that all of you listening have probably had experiences with workshops and the goal of developing a white paper from that. So next slide shows the workshop face page for the workshop that was held in 2008. And you see here the primary goal of bringing translational researchers and developers together who had an interest in marketing new diagnostic tests in this area. And FDA came and talked about our experience with the few tests that had already been approved and shared what we could in those areas, and NCI talked as well. And next slide gives us the expected deliverable from that workshop, which was this workshop report. And we wouldn't be surprised by the time frame from this that, it can take a little while to get a workshop stood up and a year and a half to get a paper submitted and finally out there in the public domain. But here's what's particularly unique about this space. It's not only did NCI and FDA work together to organize this workshop and deliver the workshop report as desired, but at the workshop, the realization was communicated to the community that there would be significant benefit in working together in the area of mock submissions to better help the community to understand and demystify the process of bringing these kinds of devices to market. So at the very same time of the workshop, 2 mock submission efforts were spawned. So next slide, please. This just gives the names of these 2. Why 2, you might ask? Why not 1? Why not 5? Well, of course, there's a balancing of the effort it takes to do these kinds of collaborative efforts and the value to be gained from them. So it's really a matter of just working together as we are with our MDIC working group to develop teams that have a shared need for information, figure out what kinds of questions would be best to be answered. And in this case, it just happened to be that these were the kinds of applications that allowed for FDA to communicate the kinds of information we thought would be of value to the community, and NCI in taking the lead, and the teams appreciated the need for the community to receive. So next slide, please. So what is a mock submission, nuts and bolts? It's a submission to the agency that is meant to represent what would be incorporated into an actual pre-market submission. But of course, because it's mock, we can focus in our attention on those sections that we think are really important, both for the community that would be the submitters as well as FDA as responders. So in the case of this proteomics example, these were the 6 sections submitted. Intended use, which is always very key for driving the classification of a device, and working together, the submitters of these 2 mock submissions had to come up with intended uses for their hypothetical devices. They also had to develop a device description, and that is also a great homework assignment to figure out, just what you have to, how do you describe a device, and how much do you describe, to what level of detail. Then they described the hypothetical analytical studies that were done on this hypothetical device to characterize it, and then finally came up with a clinical and statistical evaluation proposal. So next slide, please. And this is the punchline, that at the very same time that the workshop paper came out, this is that same face page for the white paper volume from clinical chemistry, out came the documents from the mock submission effort. And there was an intro paper that served as examples of review comments to the proteomics community and talked about the lessons learned. Not only was there a paper that, described the ongoing, process of working together, but full supplemental materials were printed that included the actual filing of these mocks, the mock filing packages, and the verbatim FDA review memo stack. So it's just this incredibly detailed approach to having a communication between industry together coming into the agency, and the agency responding widely so that the entire community benefits. So next slide, please. So here are the project outcomes that came from this Interagency Oncology Task Force Molecular Diagnostics project. Mock submissions were published together with FDA comments, as well as this overarching publication on the process that gave useful background information for proteomic device developers. So what's really important here as we work together as MDIC is to recognize that while MDIC members are working together on mock submissions, the entire community will benefit from the outcome of this work in terms of making what comes from this process widely public and available. Next slide, please. So let me just make a few comments regarding some additional considerations. In the case of this proteomics example, NCI, the National Cancer Institute, was the sponsor or submitter to the FDA. So they managed the process across the proteomics community, managed conflict of interest concerns, and were really central in choosing the submission content. There are other models for handling all of those aspects. That just happens to be the way that it was at that time. I think that the more important bullet point on this slide is that it's really essential to have FDA's review divisions on board, and we're working on that with the mock submission ideas that are coming out of our working groups. Because, of course, FDA reviewers are very busy doing their real reviews of real devices, and it's important if we're going to devote resources to a mock review that we see that it is an effort that will have the value for the agency and the community longer term in terms of helping us all to get to a place where we can anticipate submissions that are, good quality, that, based on the fact that we've communicated what our needs are, and that down the road that it moves the field forward in terms of communicating, and encouraging submissions in areas that we think are right. Next slide, please. So this is my conclusion slide. I hope that I've given an appreciation for how mock submissions can be a means to building communities. That is not only relationships among submitters, in our example in the past, the proteomics community, and in our example with MDIC, we're very much seeing relationships being built amongst the modeling community. So, amongst those folks, as well as developing relationships with FDA. It creates transparency, and in the example that I've presented, you've seen that we published review comments verbatim. Oops, I've just lost my view for a minute. And it can provide answers to specific questions beyond what is communicated via FDA guidance. In the case of the proteomics example, we answered questions they hadn't even thought about asking, but because there wasn't, an experience with the agency to know what kinds of things might be of importance, and so working together with us, we can sometimes that happens. And at the end of the day, move the field forward, and moving the field forward means contributing to the regulatory science space, addressing gaps in information regarding how devices can be characterized and evaluated to bring safer and effective devices to the patient. So with that, I'll turn the slide to my last slide, and that can be up while Dawn introduces our next speaker, just so that I make sure that we give acknowledgments to the folks who contributed to my materials. Thank you.
Thank you very much, Kyle. I really appreciate it. Great presentation. And I'll remind everybody that there is a chat window that you can access by pressing the green bubble. And if you'd like to ask any questions for Kyle about her presentation on mock submissions, now would be a great time to start typing in some of those questions, and then we'll take them towards the end. And with that, I'll introduce our next speaker, Tarek Haddad, and his colleague working on this activity, Adam Hines. Both are senior principals, Tarek a statistician and Adam an engineer at Medtronic. And Tarek is the chair of the MDIC working group that's working on the integration of Bayesian predictive engineering into clinical trial development and surveillance. Tarek, if you'll like to begin.
Great, thank you. So I just want to first acknowledge the mock submission working group. They've been a great group, and they've really contributed a lot to this presentation. Next slide. So my short outline is really talking about currently some of the burdens with determining clinical evidence using clinical trials, and then how we can help solve some of those burden problems using engineering models and simulations, and then finally, how we are going to make all of this happen. Next slide. So if you look at the current paradigm of clinical trials, we see an increasing cost in sort of the evidence required to release products. It's generally going up at about 15% per year, and this is really not a sustainable trajectory. So we feel as a group that modeling might be a way of sort of disrupting this curve and bringing that cost down a little bit. Next slide. And Dawn actually showed this slide, but it's interesting that currently, if you look at where modeling is used in the life cycle of the product, it's used in all different areas. But one glaring missing element is in the clinical trials, where there's a large majority of cost in the development of a product that goes into the clinical, and yet modeling is strikingly absent in that area. So that's where we feel we can get a big lift. If we can just add something to this realm, we could get a real lift in some potential reductions in cost and reallocations of funds, as well as other virtues. Next slide. Thank you. So what are we trying to do? Well, if you look at the current sources of evidence, it's made up of a combination of humans, animal bench testing, and computers, computer modeling. But humans really are the most expensive element. So we really would like to disrupt the whole clinical trial design with what we're calling the addition of virtual patients. And you can see we want to create a new pie wedge with these virtual patients, which would augment the real patients. And these virtual patients, now, when we start using the term virtual patient, some people get this idea of a sort of a total virtual person walking around with hair follicles. We're not thinking about it in that sense. We're using the term virtual patient to align the vernacular between clinical, statisticians, engineers, regulatory science, so we can all speak kind of the same language. And the use of the term virtual patient sort of helps with that conversation. What we mean when we say virtual patients, usually it's related in the medical device as a local effect, something like such as like a stent, and how the stent relates and interacts with the body, and some outcome of interest such as the stent fracturing or becoming non-responsive, and how that local effect happens. So something very simple with like a physical model injected with probabilistic modeling that simulates a clinically relevant outcome. And just a couple of examples are lead fracture, MRI heating, dislodgement, lead sensing. What we want to do then is take those virtual patients using Bayesian methods to incorporate those virtual patients into the actual clinical trial. By doing this, we can really maintain some important clinical endpoints, but we get some of the other virtues such as reduced sample sizes and some other attractive features. Next slide, please. One of the other interesting elements of this method is the ability to weight the number of virtual patients really as a function of the maturity of the model. So suppose you have a model that's really mature. There's a lot of validation that's gone into it. The model is something that's considered an international standard and has good predictive qualities. In a situation like that, you may really depend heavily on your virtual patients or the model. And much of the clinical evidence would really be focused on those virtual patients. In contrast, you could have a model that's fairly new, right? It's a new product, and a company wants to invest in really understanding that product. So they want to invest in their modeling, but they want to see that there's some sort of benefit to that. And if you look at the current paradigm, you generally can't utilize those models until they reach a certain level of maturity, which makes it challenging for a company to sort of see long term and say, I really want to invest for the future. With this method, it would allow you to, even though you don't get you don't use the model completely, you can still augment a clinical at the beginning stages with some virtual patients. And then as the model matures and you run multiple clinicals validating that the model gives you similar results as the actual clinical trial, you slowly can increase the number of virtual patients. And this has a much more smooth and gradual transition versus this binary effect that we are in currently, where the model is not mature and then boom, it's mature enough and now you don't do any clinical trial and it's just the model. And that transition can be uncomfortable for both the manufacturer and regulatory. Next slide, please. So we're talking about these virtual patients. What makes a virtual patient? Well, we do a lot of physical modeling, mechanical models, electrical models, heating, diffusion, understanding the device and the tissue interaction in local areas. But one of the really amazing things about a clinical trial, at least as a statistician, I really appreciate a clinical trial, is it really brings together all of those uncertainties related to patient demographics, ages of patients, all of these factors that create this natural variation. And it also incorporates uncertainties in terms of sample sizes and biases. So if we're going to really create a virtual patient, we can't just use a physical model. We have to inject some of these same virtues that a clinical trial has, these probabilistic features. So we have to understand the variability of age and gender and activity level and implant factors in order to really get a good predictive model. And not only that, but our predictive models have to be in the same language or the same endpoints as our standard clinical trials. Because a lot of times you'll see a predictive model and it'll be in terms of something like a safety factor. All right, so we've got a safety factor of 2. Well, as a statistician, as a clinical researcher, it's very hard to determine what a safety factor of 2 means when this is going to be implanted in 10 or 100,000 patients. So we need to take the models to the next level of taking it to like a time to event or a rate of occurrences, something that is clinically relevant that you would see in a clinical trial endpoint. So most of the focus in the early stages have actually been on safety and reliability related endpoints, such as nitinol, frame failures, housing cracks, MRI heating, atrial tachycardia detections, inappropriate shocks, things of those natures that really affect the safety of the device. Next slide. So I'm going to go through just a quick example to sort of ground everyone on an example that might be able to utilize these methods. And it's related to predicting of ICD lead fractures. So an ICD lead creates a shock when the person goes into fibrillation, and there can be cases where these leads can fracture in the shoulder region where your arm is moving, and this can have clinical consequences. So we actually did this case study, and we published it in the Journal of Reliability, Engineering, and Systems Safety this year. And this is a good application for this method. There are many, many other applications, but because this one is in the public sphere and there's a publication on it, it's a good example we can use. Next slide. So, in the case of a lead fracture prediction, what you need to do is you need to understand how the device and the body interact. So in this case, we collected many use conditions and did high-resolution imaging to see when a person moves their arm, what kind of stress is applied to that lead. And not only that, we had to do that on many different patients to get the whole distribution of what the possible stresses could be across a range of demographics of age and body type and activity levels. And then not only that, but we have to understand how a patient moves. So just because they're implanted in a severe case where there might be a lot of damage to the device if they move their arm, they may not move their arm very much. They may be an old, frail person that really is not moving their arm, and you need to have both of those combinations to really get a failure. So we have to understand the activity level as a function of our implant population. And then finally, we need to understand the strength of the device to withstand these types of stresses. And so there's variation from device to device. As much as a manufacturer wants to control their devices and make homogeneous devices, there will always be variation between devices. So we need to understand that. And so the methodology is really to basically take all of those factors, randomly grab a patient, and randomly grab a device and an activity level, and really put those together in a blender to see how long it'll take for that virtual device with that virtual patient to fail. And that's when we say a virtual patient, that's what we mean. And we can do that hundreds and thousands of times to get a huge cohort of virtual patients and see how long it takes their device to fail or not fail. And so each one of these blue lines in this picture represents a plausible combination of patients. And the reason why there are many blue lines is because there is uncertainty in model predictions. And any good model should account for those uncertainties. And in this case, we have uncertainties because we don't image 1 million patients, right? That's very expensive. So we image a certain number of patients, and as a result, we have estimation errors. So that's why there are different blue lines to account for the measurement, different minimal amounts of information, but we can get confidence intervals on our prediction, and that represents those black lines. Next slide. So if you look at, just as a quick example, for this particular model, we correlate really well to real field performance. You can see that the blue lines are the virtuals and the red line represents real. So in this case, we would really be able to predict the performance of this product. Next slide. So now that we have these virtual patients, in this example, a lead fracture, but it could be anything, how do we take these virtual patients or these virtual outcomes and combine them with the real patients, right? You can't just simulate 1 million patients and then just, take 20 real patients and just sort of mix them together as if they were the same thing. That's not really statistically sound. So we had to develop some methods that were really able to handle this and do it in a statistically legitimate way. And that's where the Bayesian methods really lended a hand. We can use the virtual patients as a prior in a Bayesian clinical trial. And there's a lot of framework that's already been developed through the use of Bayesian statistics for utilizing historical data. But the interesting thing is an engineering model is truly a good prior, because when you look at historical data, usually it's from a different device. A manufacturer will come in and say, this device is similar to the device we're currently using. Well, if it was similar, then why do you need a clinical trial at all? So it's always been this sort of problem with logic of what constitutes a good prior. In this case, because you're modeling the actual device of interest and the manufacturer is determined based off of looking at this prediction model that they're comfortable moving into a clinical, it truly is what the definition is of a prior. So it really works well in the Bayesian sort of framework. So we utilize a method called a power prior, which essentially it was developed to downweight historical data. But unlike historical data where usually you have a limited sample, in this particular case, because we're simulating virtual patients, we can simulate millions of virtual patients, and we want to simulate millions to capture all of the differences across the demographics, which is why modeling is so useful compared to a smaller clinical, because you can't possibly get all of these different types of patient populations in. So we needed to reframe the power prior in a way that was effective, and we wanted to make sure that it was in terms of number of virtual patients. So we wanted to say, even though we're simulating millions of patients, the actual number of virtual patients will be an equivalent to a sample size of, let's say, 100. So it's easy to communicate with regulatory and statisticians that the weight of these virtual patients is going to be 100. If the total clinical trial will be 200 patients, 100 of them will be virtual patients. So that was really important to make it clear to everybody. And the methodology that we developed not only incorporated the uncertainty in the virtual patients, but also incorporates the uncertainty in the actual patients because of sample size variability. Next slide. One of the really important points of everything is really going to be how much weight or how many virtual patients can you get in a particular clinical trial. And that's, of course, going to be a function of the quality, as we spoke about in the first couple of slides, really about the maturity and quality of that model. But we also needed to put some protections in that also safeguarded in case, even if it's a mature model, that there was not some miscalculation. So what we wanted to do is we wanted to have the number of virtual patients actually be a function of how well the two, the real patients and the virtual patients related to one another in terms of how much agreement there was. So we wanted it to be big enough, we wanted the virtual patients to be big enough to where they actually increase the efficiency of the study, right? Because that's an important element. But you also want it to be small enough, the number of virtual patients, so that the real data truly does govern the outcome of the trial. So if there's similarities between the two, you want it to be a synergistic relationship, but you don't want the virtual patients to just overpower the real patients. So you basically have a self-fulfilling prophecy at the end of this trial. So it's this kind of tug-of-war effect. So what we did was we developed this loss function that essentially said if the virtual patients and real patients don't agree, the number of virtual patients drops. And this is sort of an adaptive Bayesian trial where you take interim looks at the data and sort of determine the agreement between the virtual and real. And if there is a lot of similarities between the virtual and real, well, then you get full credit. You get full credit up to a certain max, and that max is sort of developed at the beginning of the trial with discussions with the maturity of the model. And so how we did that was we developed what's called a loss function that determined the optimal N0, and N0 is really the number of virtual patients. Next slide. So what are the benefits, impacts to both the patients and the business? I mean, we want to make sure it's useful for the business, but we also really want to make sure that it's good for the patients too. And so we really want to have both. And the nice part about this method is it really does lend to both a synergistic positive relationship for both the patients and the business. So in terms of the patient, it really, in a case where maybe the device is more dangerous than expected or have more negative outcomes than expected, we are actually limiting the number of patients exposed. Because we are doing virtual patients, we aren't exposing as many real patients to a potentially dangerous situation. The other really important aspect is that in a lot of times clinical trials have inclusion criteria that don't allow you, so you'll run a clinical trial, but it'll be very stringent, right? It'll be people between the ages of 50 and 55 who have this heart class, who are of this weight, and these clinical trials are very controlled. And so then when the product goes out in the marketplace, you have very little understanding of how that device is going to work in, let's say, pediatrics or elderly, or a lot of times clinical trials have gender biases. So this methodology, because it's virtual patients, you can sort of expand that population to these vulnerable patients that don't generally get in clinical trials. And then it's also focused, you can really focus on rare failure modes, which is a lot of times really the problem. And finally, but most important, is that when you run a predictive model, when you start with a predictive model, and then you run a clinical trial, and that clinical trial gives you the same results as your virtual patients or your predictive model, you're actually a lot more confident moving into the general public than you would be if you had no idea how your product was going to perform, and then you just tested a few hundred patients, right? This really allows you to both understand the mechanism and then validate that you understand the mechanism with a clinical. So they're very synergistic in that way. And so I think that really lends to, in the end, if it works out that your model validates your clinical, it's the same way your clinical validates your model, you in the end have much more confidence that this product is going to perform the way you expect it. And that's a real virtue to this method. And of course, on the business side, it can help with reducing the length and the size of clinicals. And then also, I put less uncertainty about the product performance in the business side too, because businesses are also very, very keen to understand how their devices are going to work. Next slide. Okay, some of the other features of this method, one of the big ones is the collection of use conditions. So one of the things that some really clever people always ask me is they say, well, in your device, in your paper on lead fracture, you had to X-ray people with that device. Well, how can you do that before the device is released to the general population? Good question. So what we brought into the method was that the first proportion of the patients in the clinical will actually be implanted with the device, but not only will they be monitored for the response or the outcome of the clinical trial, but they also will be used as the input. So if in the case of a stent, you would X-ray the stent in the patient to see how the stent is reacting, or in the terms of leads, you would see how the lead is interacting, and then you'd be able to update your model based off of the true use condition or input data. And that would actually be very beneficial. And it's very cost efficient, because then the patients in the trial are not only just being used for the outcome, but they're also being used for the inputs, and that's cost effective. The other thing is this methodology is perfectly adaptable to post-market surveillance. It allows you to determine where thresholds should be much easier if you've got a model and the post-market surveillance methods start diverging from the model. It allows you to understand maybe something's happening in the field, or maybe something's happening on your manufacturing floor. So it really lends to the modeling being used in the full life cycle of the product. Next slide, please. So just to discuss really quickly the activities that are going on with regards to this method, there's a lot of collaboration with the FDA through the MDIC working groups. We're working on the FDA actually published the use of Bayesian methods, and that was a really important publication by the FDA to say you can use Bayesian methods, and so that's key. The work that's happening in the Augmenting Clinical Trial Working Group at the MDIC that was started in May 2004. There's a publication that's going on related to development of these methods, and that's a publication that's being co-authored by industry and regulatory, or FDA. And then there's the mock submission work, which of course we talked a lot about that today, and that is planned for developing a mock submission related to a device that would have a mature predictive model as an outcome, and being able to augment the actual patients with virtual patients. Next slide, please. So in summary, this methodology is really trying to bring clinical trials into the 21st century. We're highly computerized virtual society, and it's reasonable to think that these models and these in silico or virtual personalities would be something that would increase over time. So this methodology may be at its infancy, but think about it in 20 years, where modeling and simulation becomes even a larger portion of our society. And so we need to have the framework in place that can utilize these methods in our traditional clinical trials. And so the work that's being done in this area, which I've already talked about, is really building credibility for these types of models, joint publications with industry and FDA, and then the mock submission. So I think that's all I have. Thank you.
Great. Thank you very much, Tarek. And then I'll remind everybody that for the Q&A session that we have for the last several minutes, we have the chat feature that you can access by clicking on the balloon that looks like a talk bubble. We have several questions that have already been asked. And Tarek, since a few of them are clarification questions around your presentation, maybe I'll start by asking you a couple of them.
Sure.
One of the questions was incorporating, and these two go together a bit, so I'll ask these two together. Incorporating virtual patients into clinical trials. Does this mean you're using the results from modeling virtual patients as a prior? And then the other part to go with that is, and if the results from the clinical trial come out different than the model, what do you do then?
So it's a very good question. So the first question, yes, the virtual patients would be kind of akin to if they were historical data sets, and you would use methods. And so when we release our publication, which should be coming out in the next year, let's say, it'll discuss a lot of these methods, but really the virtual patients would be sort of considered prior data, so they wouldn't be right in the clinical, but they would be used as priors in a Bayesian clinical trial. The second question related to if there's divergence or non-agreement between the virtual patients and real patients, then if we can go back to my slide deck on the penalty function or the loss function, right? Up. I think it was one
Keep
more.
Going up. Oh, too far.
I'm sorry,
going
down,
faster, I think.
Down. Near the bottom, right? One slide up. Well,
I think
there it is.
One.
There it is. Yes. So this is really where we control the how much weight you get on your virtual patients. So if there is agreement, then you get quite a bit of weight for your virtual patients based off of some predetermined amount. But if there's differences, such as the virtual patients are much more show that the failure is much smaller than the actual failure, then you get penalized, and then you start losing the amount of virtual patients. So essentially, in the case where your model is completely wrong in a negative way, then what would happen is the clinical trial would convert to a regular trial, and you may fail the trial. But we have protections in place through the method that essentially you do interim looks at the data, and if there is a disagreement between the virtual patients and real patients, you get penalized in terms of the number of virtual patients, and that penalty will drop to zero at a certain point. So we do protect against that scenario. That is one of the key points of this method. A good question.
Great. Thank you very much. So I'm going to ask a question for Kyle now. The mock submission approach is very interesting. You touched upon limited FDA review resources. Was the review of the mock submission in line with FDA time requirements? And would it be included in the FDA performance goal metrics? Thanks.
Sure. It was not the case that the experience in the past contributed to FDA's performance goal metrics, and we're not thinking along those lines in our working group efforts with MDIC as well. At the same time, though, you can see from the experience that I was pointing to that, within a fairly short amount of time, after a workshop, not only was there a workshop summary developed, but that same time frame is what it took for this external group to convene, develop the mock submissions, for the response to be developed, and for the publication to happen. So it really gets to the heart of that important bullet that I pointed to, that you have the buy-in from FDA internally, that they will make time for this because they see value in it. And, as much as we do have tight review timelines, we are working with MDIC because we see incredible potential for collaborating together to support innovation and development of regulatory tools broadly.
Great.
I'm wondering,
thank you, Kyle.
I had Giovanna on the line if she wanted to say anything more in terms of the process herself, she can answer.
That would be great.
Hi, this is Ivana. So the specific mock submissions that Kyle was describing, we did, at that time, there was no performance metrics for the pre-submissions, which these were. But we did actually, I think, we would fulfill the requirements for those. So we were pretty fast with review, and I've seen the question also about whether the review was interactive. And again, at that time, the interactive review was not something that was mandatory, but the office that I'm in has been working on with interactive. And I think for these kind of collaborations, interactive review is really, really important, because sometimes the submitters of mock submissions are trying to learn how the regulatory system works in the first place. So you do need to have a lot of interactions and try to collaborate and help the process move along. Is that helpful?
That's great to know. Thank you. That was very helpful. Thank you very much. I'm going to go back to, I'm going to take a question for myself shortly, and then we'll go back to Tarek with a couple of questions. There was a question, data was presented regarding the use of CMS in different phases of development with no use in the clinical or reimbursement phases. And on the major barriers to more use of modeling and simulation was uncertainty in the regulatory expectations. Is it possible to put these two together? That is, in the areas of CMS where it's already widely used, what has the regulatory experience been? And as a program manager for the Modeling and Simulation Project, I would say that we have each one of our working groups working at a different piece of either barriers or opportunities. And so we're very much working to see how we can put these together. And when we can put them together, we do so. And when we can't, we try to diversify and attack the question on multi-fronts. So I think that was great. And then, Tarek, the question to you was a bit around models. Are you already using disease modeling, microsimulation, population modeling, and techniques to support what you call virtual patients? And if you do, are you using it in design, analysis, tracking, validation, where in the product lifecycle?
Okay, so we do use modeling in many areas. There's a large, at least at Medtronic, there's a large team of people running all types of different models. Some of the questions related to disease modeling, those are related to efficacy versus safety, and those models are being done. Those models are perceived as harder to do than device reliability models, but nonetheless, yes, Medtronic and I'm sure many other companies are working on efficacy and disease state models as well. And so in the future, I see that those being used for this methodology just as often.
Great. So disease modeling is kind of one of those forward-looking activities that can really get to some of the efficacy questions. Certainly, we can also answer safety questions. And in all of these virtual patients, population modeling techniques are really important. I think that was kind of what I took from your answer. To follow up on the disagreement question, when your population size, this will be our final question, then we'll conclude. When you use this Bayesian prior method, would it be necessary to increase enrollment numbers to ensure statistical significance?
Yes, it would be. Depending on your adaptive design, you may find if you define your endpoint as not being futile yet, let's just assume that your model is not performing, the real patients are not performing the way the virtual patients are, you'd have to make decisions about enrolling more real patients to supplement the virtual patients you don't have anymore, or define the whole study futile, that your prediction was wrong and the whole product is not performing the way you expected. So those are the decisions you'd have to make, of course, and those would all be clinical design values and parameters, and that's what we'll probably get into a lot in the mock submission.
Great. And would that be similar in the total number of patients to a typical non-Bayesian clinical trial, or would it be smaller or larger when the disagreement occurs?
Well, it would ultimately converge to a completely traditional trial. Probably end up being more slightly more. That's a tough question. It's hard to know
It's
exactly
in the details, but it's similar to great. And Kyle, would you like to say any concluding remarks as we close the meeting?
I just am appreciative of being able to speak in this first MDICx series, and I am happy to take any questions afterwards through you, Dawn, regarding the approach that we have used in the past and look forward to using again to work collaboratively with our stakeholders. So thank you.
Thank you very much. And Tarek, any closing messages?
I just echo the same things. If anybody wants to contact me with questions, I'm happy to answer those. I'm sure there's a way to find my email at some way, or you can just contact Dawn.
Absolutely. So thank you very much, everybody. We'll have a post-meeting survey, and we'd like your input. Have a wonderful afternoon.
Thank you.